Yes, so I'll be talking to you about the work that we've been doing in Mexico for upscaling coral restoration. As we know, we're in a changing climate, so this is getting even more and more important. Basically, the outline of my talk is first we'll talk a little bit about coral cover loss in the Caribbean and what's causing it, and the status of some reef building species. I'll be focusing on one species in particular, although we have worked with a number of species. Talk to you a little bit about coral reproductive biology and ecology, and then using restoration as a strategy to improve coral cover, and basically end with the upscaling techniques that we've been developing in my lab. So first I wanted to sort of go back to last year and the sort of what caused quite a lot of press was the whole biodiversity crisis numbers that the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services published, where they talked about basically 1 million species being threatened with extinction within decades. And just to put that into perspective, when I looked up the total estimated number of animal and plant species on Earth, that's about 8 million. So we're talking about 1 in every 8 species. And so in the wider Caribbean, where we have 65 species of corals, we're talking about a potential loss of about 8 species, let's say. But during my talk, you'll hear that this number is actually going to be a lot higher. And also the average abundance of reef-forming corals in the Caribbean has fallen by at least 33%, and that number is probably going to be a lot higher in the next couple of years. So the drivers of this change, there's basically, IPBES said there were 5 basically large global impacts. The first is changes in land and sea use through urban development, through agriculture and mariculture, as well as runoff and sedimentation. The 2 photos that you see there, the one on the left is Cancun, the hotel zone. So this was taken in 1970. You can see it's basically a bunch of sand and some mangroves. There's a lagoon up on the top of the photo, and then basically the Atlantic Ocean towards the right. In the next photo, this is 50 years later, and basically that whole area, it's basically a 25-kilometer strip. It's just full of hotels. So you can imagine the stress that this kind of development, tourism development has on both the ocean and the lagoon, and especially in terms of eutrophication. The second global impact is considered to be direct exploitation of organisms, so basically overfishing. The photo that you can see there is basically a big boat that's full of conch meat. This is one of the biggest mollusks in the Caribbean called Strombus gigas, and it's well known for conch fritters and things like that, and it's basically been overfished. And this is just sort of one boat in the 70s just fishing out just thousands of tons of this species. And so it's now close to extinction. The third is climate change. Bleaching events, you're all very much aware, I'm sure, of what's going on in the Great Barrier Reef with the successive bleaching events from year to year. In the Caribbean, we also have had bleaching events, not necessarily as severe as in the Great Barrier Reef, and not as successive, but there still have been impacts of bleaching on these reefs. And then, of course, there's pollution from nitrification. The photo on the bottom on the left is a sargassum bloom. I don't know if you've heard about this in Australia, but basically throughout the Caribbean, we have a floating sargassum, which has an important ecological role in, for example, baby turtles, when they're swimming out in the ocean, they'll settle onto the sargassum just to rest from swimming and just to have a sleep. And there's also a lot of organisms associated with sargassum, but due to a number of different factors, the sargassum has just increased in volume. And so you can see these white beaches are now basically these sort of brown, very stinky beaches, and the water turns a brown color, and it's basically just full of lots of nutrients, which then, of course, will get onto the reef and cause algal blooms. And then the photo on the right is basically plastics that are sort of caught up in amongst corals. So, you know, plastics are becoming a big issue as well in terms of, well, not becoming, they are a big issue in terms of pollution. And then the fifth major impact is considered invasive alien species. So, for example, the lionfish, which you're all probably familiar with from the Pacific, but it has invaded into the Caribbean and is a major competitor with many other fish species. One thing that the IPBES did not bring up as a driver of change is diseases. And at the moment in the Caribbean, there is the sclerotinian coral tissue loss disease, SCTLD, which is affecting approximately 20 species. And just to give you an idea of just how rampant this species is, this is on the left side where it says January 2019, we have a picture of pillar coral, Dendrogyra cylindris, which in this case, it's 4 meters high and about 4 meters wide. It's a colony that's probably thousands of years old. And within 3 months, by April 2019, the whole colony had died from this disease, and it's not coming back. So this is not the only disease that has affected Caribbean species, and especially this disease in particular. It's affecting, like I said, I think it's about 23 species now, and some of them are coming close to extinction. Dendrogyra is one example. So in the 70s, we also had this white band disease. You can see that this white band on the photo on the right is basically a disease that was affecting this species called Acropora palmata. In the photo on the left, you can see that this species is really amazing architecturally. It has this beautiful three-dimensional branching design where many fish and many other organisms can use it as a habitat and a refuge. But in the 70s, this disease wiped out, and you can see basically how quickly this species lost cover in the Caribbean. And so basically what we're facing now is sort of a flattening of coral reefs. So instead of having this gorgeous three-dimensional architecture where a lot of species can hide, the species that are sort of replacing these branching species are sort of flat, encrusting species, and so they don't provide much habitat or refuge for the biodiversity in these areas reduces. So basically what we did was look at the status of this particular species, Acropora palmata. It's also called elk-horn coral. We looked at its population status in the Mesoamerican reef, which runs from the northern part of the Yucatan Peninsula down through Belize to Guatemala and makes sort of an L shape in front of Honduras. And so the yellow points that you can see in the graph on the left are places where you would expect Acropora, or there once was Acropora, but now there was no cover left at all. Whereas the blue where we did get some coral cover, and those blue points are then shown in the graph in the inset, where you can see Acropora palmata cover, basically reef site. So the issue is there are a number of values that are very close to zero. There is one value that's quite high. It's in around 35 or so % average, which is a site called Limones. It's a site that's very close to where we work. We've done a lot of work. And because of this publication, that reef actually got close to fishing and to tourism because it was seen to be such an important site for Acropora palmata along this whole Mesoamerican reef. And so basically the issue here is that even though there's been a lot of management effort in terms of making national parks and having restrictions on fishing and on tourism, etc., there's been no significant recovery at all of this species. And also recruitment is really low. Basically, nobody has been able to find much recruitment at all, mostly because the reefs are overgrown by macroalgae. Young recruits tend to be preyed upon very easily. Also due to increased temperature with sea surface temperatures being higher, that could possibly lead to the recruits not surviving. There may even be also more inhibitory crustose coralline algae. We now know that CCAs, as they are also known, which tend to promote coral recruits settling, may actually, there are some species that may be inhibitory. And also due to pollution and both in terms of nutrients and in terms of sound, recruits basically do not, or the larvae just don't want to settle in areas where the smell is strange, like on degraded reefs. Apparently the smell is different to non-degraded reefs. And the changes in sound, because there's a lot less, I guess, sort of good sounding organisms that are making noise in the reef, and so the larvae tend to not settle there. So the problem then, of course, if we don't have recruitment and we have fewer colonies, there's less chance of fertilization between the colonies, and then that means there's lower genetic diversity, and so there's a decreased probability of surviving climate change, because we know that genetic diversity is very important in terms of being able to survive climate change. So all of these factors together mean that Acropora palmata is on the critically endangered, on the IUCN Red List. And I'm actually on the committee now to reanalyze all of the Caribbean corals for IUCN, and unfortunately, I'm pretty sure that a lot more species are now going to be added to the red list as critically endangered because of this disease event that we're having at the moment throughout the Caribbean. So that's basically been on the red list since 2006, I think. It's covered by the screen, but and also since 2001, the Mexican National Park System has protected this species as well. So just to give you a little bit of information about why we sort of focus on this species, it's a pretty fast grower, especially in shallow water, and it's very important on reef crests. It's a major reef building species. In fact, in my opinion, I think it's one of the most important coral species, not just in the Caribbean, but in the world, due to its importance in reef accretion. It forms a very solid base and then produces these very tall, three-dimensional structures that can house a lot of different species, including commercially important species such as lobsters. It also plays a unique role for coastal ecosystems because the species grows on the crest and is so strong, it has such a strong skeleton, it absorbs up to 99% of wave energy. And because that wave energy doesn't reach the beaches, it prevents beach erosion. So during hurricanes, when you actually get a lot of wave energy coming in to the beach, the presence of reefs that are dominated by this species mean that not only the beach, but all the infrastructure that's close to the beach is then protected by this species. However, we're seeing more signs of degradation. So the photo on the left is the Limones reef, the one that has the really high Acropora palmata cover. And this is a reef just 2 kilometers south of there that once used to be dominated by Acropora, but now you can see it's very much degraded. And basically, this is one of the sites that we're working on for trying to restore. So basically, one of the only things that we have left to do in terms of conservation is to start restoration programs. And basically most programs, especially in the Caribbean, are designed to increase biomass and increase the number of colonies, principally through fragmentation. But we're also interested in trying to produce self-sustaining populations, and basically through using assisted fertilization and larval husbandry, and that way we also enhance genetic diversity. So then the idea is to sort of fill in these gaps between these very dispersed colonies so that they can reproduce amongst each other. So we need to consider several factors. One is the coral biology and also the challenges. So in general, people feel that we need to have a lot of expertise for this, and that's very costly, and that the problem of scale is so big that, you know, why should we even attempt this? So basically, we've taken on these challenges and have basically tried to show people that you need a little bit of knowledge, but really it's sort of like cooking. You know, if you just like to mix some ingredients and just be a little bit careful, you can grow a lot of coral recruits. And we're also finding ways to reduce costs and also to upscale. And basically, the keys to success for us have been to do solid research and to collaborate with different organizations, to get involved in capacity building. Those are the three topics that I'll cover mostly today. And also, in our opinion, community involvement and outreach are also very important, but I won't cover those topics today. So in terms of coral reproduction, and actually my team is out tonight looking for spawning, because there's one species that might be spawning tonight. And basically, it's an event that happens in the summer. Tends to happen in the summer. It can happen in the spring as well. But basically, on 1 or 2 nights a year, mature colonies will release packets of eggs and sperm. That's the sort of snow-like vision that you get there, and the word under the word spawning in that photo. And basically, these packets of eggs and sperm will rise up onto the surface of the water. They'll break up. And then because different colonies have spawned at the same time, they tend to be quite synchronous. There's then just basically an orgy going on at the surface. A lot of fertilization happens between the colonies, and then you get embryonic development. And then you get, which is shown at the top, and then going around in the circle at 2 o'clock. Is the larvae that are formed within a few days, and that's the dispersal stage. So this is all of this is happening at the surface of the water column. So during these 2 or 3 days until the larvae are formed and start to swim and start to look for a place to settle, these are being moved by the currents. So then, I think a pretty amazing event happens. These larvae, which are about 2 or 3 millimeters long, will somehow find a reef, say 3 or 4 meters depth, and will somehow find a place to settle. And they undergo a process called metamorphosis. So they go from being a pelagic organism to being a benthic organism. They'll basically sit with their butt down onto the substrate and will metamorphose into what's called a primary polyp. That's the photo that you can see sort of at 4 o'clock. And you can see that it's getting a brown color because it's taken up symbionts and has formed a mouth and tentacles. And you can also see a skeleton that's begun to form. So then that primary polyp will undergo reproduction and form a juvenile, which you can see at 6 o'clock. And then if we go around to 8 o'clock, that colony will grow into a mature colony, which will then spawn after about 3 or 4 years. And, sorry. So then basically we take advantage of this spawning event to collect spawn and the nights when we predict the spawning will happen. And I'll talk a little bit more about that later. But there's also another way that corals will reproduce, and that's through fragmentation. So through a storm or through jet skis or a diver kicking a branch. Basically, the coral will break into different pieces. Some of those pieces may survive and form a new colony. So in the photo at 8 o'clock, where you can see there are two colonies, one a bigger one on top and a smaller one on the underneath. There, we're not sure if those two colonies formed by fragmentation or if they formed from two different individuals that settled in those places. And so in terms of reproduction, that's really important for us to know, because if we collect spawn from those two colonies and they're the same genotype, they will not fertilize. Fragmentation is used by a lot of different groups to produce more colonies to increase biomass. So it's a very popular way to do restoration in the Caribbean, at least. And just to go back to the symbionts, the symbiont of Acropora palmata is called Symbiontium fiti. It's not inherited from the mother to the offspring. So the offspring, once they've settled, have to take up the symbionts from the local environment. And as I mentioned earlier, because the larval stage is the dispersal stage, the larvae may actually settle a great distance from the mother colony. So if we're growing these up in culture, we then need to provide the symbionts through seawater or by having fragments of the colony nearby, so that the symbionts that tend to get expulsed or expelled will then get taken up by the sexual recruits or the primary polyps. This species is highly phototrophic. It really depends on symbiont photosynthesis rather than heterotrophy. It's a shallow dwelling species, so it has a lot of light. And so obviously once the symbiosis is established, they need to have light. So this needs to be taken into account when we're outplanting these recruits. So we've done a little bit of population genetics because we do like to know, which genotypes we're dealing with. Are we dealing with clones, or are we mainly dealing with sexual recruits? And so basically we did a study along the Mexican Caribbean. And what we see is that there's no genetic differentiation between the sites. I think we did something like 9 sites along the Caribbean. So it's basically one population, and shows that we have very high connectivity in gene flow. This wasn't a surprising result, because within the Caribbean, especially the Western Caribbean, that is the case, where there's high connectivity in gene flow for this species. But in terms of the implication for restoration is that theoretically we can take gametes from any of these sites and move them to other sites without affecting gene flow. However, the story for the symbionts is different. We did find 5 different clusters, and basically, so there is a genetic structure based on isolation by distance. So this is, for us, really interesting, because in terms of restoration, the implication is that when we outplant spat, the sexual recruits, it's probably better to outplant them prior to symbiont uptake, so that they can take up symbionts locally. Because just remember, with the dispersal, they can be very far from the mother colony, so the light conditions might be different. We also, because we were able to basically get the sort of first and last name of every single colony that we were able to sample, we were able to do sort of clone mapping and show that clones could be as far apart as 13 meters. And this is a really important result, I think, because a lot of people will say, oh, I'll make sure to collect my colonies at least 5 meters apart to make sure that they're genetically different. And in reality, because we find that clones can be, the same clones can occur up to 13 meters apart, then it means that we have to be really careful when we're doing our reproduction work, when we're gathering spawn, to make sure that we're collecting from genetically distinct colonies. And we were also able to do a series of different physiological experiments, etc., which I haven't included here, but we also did some work on susceptibility to fragmentation, and basically showed that about half the genotypes, I think we've outplanted 8 different genotypes, and half of them were basically all died from like single genotypes. So there are certain genotypes that are just not able to withstand breakage as well as other genotypes. So then basically going to the spawning work that we have done and how that sort of works in with the restoration. We do focus on Acropora palmata, but we have worked with other species. And basically what you can see there in the inset is this, is the sort of bundles of eggs and sperm in the polyp mouth just before they're released. And then on the bottom, you can see that the nets that we use to capture these gametes, and then we fertilize them in the boat, and then we bring them back to the lab and then basically culture them. And you can see that the lab is full of people because we get people from all around the world coming to work with us in workshops and in graduate courses to learn how to do this. So one of the ways that we upscale is by having many different sites. We've worked in up to 11 different sites in some seasons, going from Mexico through to Belize. Some of these sites have, 2 or 3 different groups working, so it means that we can really increase the genetic diversity of the sexual recruits that we produce. And we've also been working with quite a number of different species. We've increased up to working with up to 8 species at a time. Fortunately, they do spawn on different nights, some of them. Sometimes we get 2 or 3 species going up on the same night, but we basically, with working with these different species, we can sort of extend our spawning season. And another way that we can increase genetic diversity is through cryopreservation. So we've been producing a sperm bank to basically freezing sperm so that we can then, on any night where we may not have a very good spawning night, we can then basically defrost some sperm to sort of increase the diversity that we're working with. We're also working and trying to freeze viable eggs. We've gotten to a point where we can freeze the eggs without them exploding, but they don't yet fertilize. So we're still working on that. And also on freezing and culturing primordial germ cells, trying to find ways to be able to culture more colonies. And basically we have had successful fertilization, embryogenesis, settlement and growth of Acropora palmata and also the brain coral, which is called Diploria labyrinthiformis. That's a species that hopefully is spawning tonight. And we've done that using frozen sperm and have been able to outplant them onto the reef. So in terms of our results, we've been trying different techniques. The first techniques we tried was doing long-term ex situ culturing. Basically, we would produce the larvae in our lab, settle them onto substrates, and then we teamed up with people from the Xcaret Aquarium. This is basically a tourist company that have a really amazing aquarium facilities. And so we basically did outgrowth there in their aquaria. And we then, once they sort of grew up to juvenile stage, we then transferred them to in situ nurseries. And then we've outplanted 70 of these colonies onto reefs that have been damaged by ship grounding. And we basically outplanted them at about 1 colony per meter squared. And these colonies are now reproducing. I don't know if you can see there, but there are these sort of little pink colored balls within the that white skeleton that's been exposed, and those are basically the gametes that are produced by the corals. So these corals are now reproducing. However, the cost per year doing it this way turns out to be about $45,000 US, which for countries in the Caribbean is just way too high. So basically, the problem we're facing is that restoration we need to attend on a hectare scale, not a meter squared scale like we have been doing. Basically, our objective was to produce or to develop cost-effective large-scale methods by limiting handling time and by decreasing labor costs. So instead of waiting 2 or 3 years to outplant these corals like we were doing earlier, our approach is to try to seed them at about 2 to 4 weeks post-settlement. So obviously this cuts out of a lot of costs of maintaining them in aquarium, maintaining them clean, and all the labor costs that are associated with that. So basically, it starts with large-scale gamete collection. For example, working at 11 different sites along the Mexican Caribbean. We've also tried developing the embryos in floating ponds, like you can see in the middle picture there. It doesn't really work very well in Mexico for 2 reasons. One, we have a very open coast, so these floating ponds tend to not do so well. And also because of the sargassum blooms, and there's so many nutrients in the water, it really does not work very well for us. But also because we're upscaling, we need to condition a lot of substrates. Instead of hundreds of substrates, we're talking about thousands or tens of thousands, and maybe even eventually hundreds of thousands of substrates, and they weigh about 100 grams each, so it gets pretty heavy pretty quickly. So we need to work out how sort of an engineering feat of how to deal with this. And the reason that the substrates need to be conditioned is because a biofilm needs to form on the substrates for the larvae to settle on them. They really like to have this sort of bacterial biofilm. So this is an example of one of our substrates. It's called a tetrapod that was developed by Secor International, which is one of our collaborators. These are self-seeding or self-attaching substrates, so we don't use any cement. There's just this, the 4th pod is in sort of a crack in the reef. And all those little white dots that you can see on the substrate are the primary polyps that are about, in this photo, they're about 4 weeks old. So we basically outplant them, and then after a couple of years, this is about 2 years later, this is an Acropora, on the bottom photo, you can see an Acropora palmata colony that has grown up, and underneath there is a substrate. So we make sure to monitor all of the outplants just to look at survival and growth, and try different techniques to outplant. The cost of this is about $15,000 per year. And one of the major challenges that we have is called the post-settlement bottleneck. We do have very high mortality in the coral recruits, mainly. Sorry, I'm not sure why that happened. So mainly there's algal overgrowth, as you can see in the photo on the right, and we do get some of the colonies simply just don't take up symbionts. So then we've worked further with Secor International, looking at different microhabitats on the substrates. So we have canals, and we have protuberances, and we have little sort of indentations, and all different kinds of shapes to see which once they've been outplanted onto the reef, which actually survive better, which don't move, and which microhabitats work best. So far it looks like the little canals are the ones that work best, where the corals will settle down within the canals, and I'm guessing that the predators can't really get to them, because I think that's the sort of major problem we have with predation when they're very small, especially with grazers, sea urchins, and things like that. And then another way that we have been upscaling is to improve our culturing facilities. So we basically started off with tiny little containers and then worked our way up to 1 liter containers, and then to, I think these are about 40-liter containers on the bottom. And now we have this culturing facility, which is actually 2-story, and we can handle thousands of substrates at a time. And it's a really simple design. It's really easy to use, and yeah, it's very user-friendly. All of my technicians are very happy that we have this new system in place. And we've also developed mobile laboratories for culturing corals. So this is a design we started using in Belize in a collaboration with the University of Belize. And basically, it's really simple. It involves, on the left-hand photo at the top, is basically a reservoir. That's where the seawater is held within a reservoir, but that reservoir is buried within sand. And the beauty of sand is that it actually maintains temperature, seawater temperature, about 27 or 28 degrees Celsius, which is just perfect for growing coral larvae and doing larval husbandry. And we have on the photo in the middle, on the top, is basically, it starts with a UVC filter to just take out any organisms, and then a series of filters to take the water, basically to filter down to 1 micron. And that's important in the first 5 days of culturing. And then basically just this very easy sort of culturing system. You can see in the middle at the bottom a photo of one of our tetrapods, which has lots of Orbicella faviolata, which is a big mounting coral settled on these substrates. And the cost there comes down to about $1,500 US. So, we've really been able to reduce the cost. And so this means that, even in really remote areas, the Calabash Key Field Station is about 2 hours steaming time from Belize City. So it's actually worked out really well for us. And also another way that we've been sort of upscaling is trying to teach as many people as possible using our techniques. So far we've had about 150 participants in 10 or 12 workshops that we've run, and graduate courses from about 15 countries. And we sometimes give the courses in English, sometimes in Spanish, depending on who's coming to the courses. And from that, we've basically built sort of a pool of people who have been working with us and have now leading programs in other countries. Within Mexico, we have about 3 different groups that have learned with us. We also have a group in Belize, in Costa Rica, in Colombia, in Cuba, in the Bahamas, the US Virgin Islands, and the Dominican Republic. And so basically with these workshops and graduate courses, they're hands-on. We do teach about the theory, but the real focus is to get out in the water to collect, spawn, and to learn how to handle it and how to do the whole larval husbandry process through to settlement, and then also teach how to outplant and to monitor these. So basically that's the end of my talk. All of this work is obviously thanks to a really wonderful group that I call my dream team. Some of these members have now gone, and there are some new ones that have come in, but basically all of the work that I've presented here has been thanks to this amazing group of people. And also just want to thank my university and the postgraduate division, also to Alina Schmant, who back in 2007 came to my university and basically taught me a lot of the techniques. And from there, we've basically developed this whole restoration program. I've had funding from the Mexican government, from different federal agencies, also from different NGOs and businesses, as well as several aquaria from the United States. So basically, I'd just like to end saying there's an African saying that it takes a village to raise a child, and to do reef restoration will take many villages, in my opinion. So that's basically all I've prepared for you today. That's my email if anybody would like to contact me, and on Facebook we have a page called Corallium La Vida de un Coral. Thank you.